joining today's event. This is the first time we've done this type of event. Instead of a traditional workshop with presentations, we have three breakout rooms operating at once on three different topics. We have guide RNA design, vector cloning, and plant transformation. And I'd like to introduce today's speakers very briefly. So speaking on guide RNA design is Colby Stalker and Erik Myers from the University of Minnesota, and Vina from the Donald Danforth Plant Science Center. In the second breakout room covering vector cloning is Josh Clem from the University of Maryland and Kien-Sub Lee from Iowa State University. Lastly, Wayne Parrott from the University of Georgia is covering plant transformation. I'm so grateful to all of our speakers that they're here to share their expertise with us. I'm sure you feel the same. So if you'd like to learn more, you can visit our website and read their biographies. So in a minute here, I'll open the breakout rooms. You are encouraged to move between the different rooms so that you get information on all three topics. You should be able to exit a room and come back to the main room, or you can move directly between the three breakout rooms. Once you are in a breakout room, you can either raise your hand to ask a question using the toolbar at the bottom. There's a raise the hand function. So you can raise your hand to ask a question, or you can type your question in the chat. All of the breakout rooms will be recorded and available online.
That web-based tool, it helps me kind of figure out where within a region of a gene that I'm actually trying to target. I think maybe before that, though, what you really have to do is understand your gene. This is one of the things that I think everybody who works in this field very often we get asked this, and we always want to say like, okay, well, tell me more about your gene. There's not like a straightforward answer to answer every single question without knowing a lot more about the gene that you're working on, because we don't know, the people who are trying to help people design guides, like um, how many homologs there are, how close those homologs are within the genome, the actual functional significance of the gene, whether or not it may or may not be lethal, whether or not there's something like, say, dominant negatives that exist in the literature. And so those kinds of things, you need to know about your gene before you can sort of say, okay, well, I'm going to design a guide RNA in one area. Typically, and probably for the purpose of this presentation, most of the things that we're going to address are going to talk about knockouts. So basically, simple frame shifts, which we consider to be simple kinds of edits. Basically, what I do if I'm trying to make a knockout is I usually design two guide RNAs for the same gene that are in the coding sequence, that are somewhere in the kind of 5' end of the gene, or will hit an area of the gene that is known to be extremely important for function. So those are kind of the first things that I look at. And then I use one web-based tool that I like, and I'll talk a little bit about that. I'm sure people will have questions. I know Erik uses Benchling to help design his guides. I actually don't know very much about using that. So Benchling for guide design, he can probably talk about the pluses and minuses of that. Um, but really, one of the things, so the tool that I use is shown here, which is CRISPR-P. I think it's actually called CRISPR-P 2.0. The reason that I like this one is that you can really see a lot of the potential off targets. You'll actually see.
Colby, can you put your slide in the presentation mode,
Sure,
please?
absolutely. Is that working?
Yeah, thank you.
Okay, cool. Yeah. So this is CRISPR-P. And so this is just a screenshot, so it's not a live image. But as you roll over these guides, designs, it pulls up this kind of information, which tells you basically off targets, potential off targets. I like it because it highlights the nucleotides that are different in red. It tells you the number of mismatches, the locus, and the thing that I really like about this tool is it tells you where those potential off targets are in the organism in which you select to design these guide RNAs. So, if it's an exon or intron, basically, or intergenic, that's basically the three kind of categories they have. And so I like that part of it. They do give you a score, an on-target score. I mostly pay very little attention to those on-target scores. If I have two or three guides that are equally do everything that I care about, and the off-target potential off-targets are about the same, then I usually do go with the one that has the highest score. But generally, I'm not terribly convinced that those on-target scores mean terribly much. Uh, sorry, I'm going to skip a little bit. Um, and I just want to talk a little bit about why the sequence of the guide RNAs, there's still so much that we don't understand. And so this is a figure that I always like to show people when I'm talking about designing guide RNAs. And this comes out of the lab next door to ours, Feng Zhang's lab, and the first author is Trevor Weiss. So Trevor, he took, and this is all in Arabidopsis. He basically found a sequence that was present 9 times in the plant genome in Arabidopsis, and then he basically monitored in somatic cells what what the guides like the efficiency of cutting was in those somatic cells, and this is all done by high throughput sequencing. And so again, this is all exactly the same guide RNA monitored at 9 different sites. And what you can see is that there are vastly different amounts of editing. So, some of this you just kind of have to test these things or try them in the organism of interest. Because, this one worked extremely well at this one site. This is reasonable, and then basically no activity at 5 of the 9 sites. So this is one of those things where we're like, we don't actually understand. I mean, at some level, we think unmethylated sites are going to be much more accessible and available for editing. But clearly, but the difference between site 8, 7, and 4, those are all basically unmethylated and open. And there's something else going on with the genome that dramatically affects the editing efficiency. So this is like always in my background thoughts when I'm actually uh, designing guide RNAs. So I'm going to stop sharing at this point, and so maybe we can start taking some questions. Unless Erik, you want to talk a little bit about benchling and how you design guides?
Yeah,
Uh,
let's go. Yeah, have Erik finish it, and then we
sure,
go back. Yeah, thank
sure.
you.
So when I design guides, like Colby mentioned, I often use Benchling, which if your genome is uploaded to Benchling, you can use it for off-target scores. Although, like Colby mentioned, I often ignore those because we just track the gene of interest usually that we care about and forget about the off-targets. But Benchling will give you an output of all of the guides and then align them to the sequence so you can see where it lands and then automatically populate a file with primers required to clone the guide RNAs. So that's kind of nice. Um, but yeah, that's everything else. I agree with Colby. When I design guides, I target open chromatin regions. I usually target the 5' end of the gene to make sure you get a frame shift mutation early on to disrupt the rest of the coding sequence. Unless you want to target protein domain of interest. But yep, that's all I've got.
Erik, can you tell for the interest of everybody who's attending, Benchling, is that accessible to everybody free or how that works?
Yeah, Benchling is free for academics. So you can create a profile and they give you like 3 GB of, or at least when I signed up, they gave you 3 GB of notebook space, file space, and whatever. So I can share a link in the chat.
I
Helpful.
have a question. Can you please
ahead.
put
Yeah.
your slide, the first slide, just a general explanation of the CRISPR that Mr. Colby, if you can do that, your presentation.
I can and am working on that.
All right, great.
While Kobe do that, when you have
Okay.
a question, please raise your hand, and then if you're open to ask your question, can you please identify your name and then affiliation, your university or things like that? Just
All right, I'm Julia
name.
Ray,
Yeah.
Cornell University.
Thank you.
All right, so this picture shows that the gRNA comes on one side, on one strand to recognize, and the other strand is the PAM region. Is that correct?
That is correct.
So when you design I designed quite a few of the CRISPR constructs, and they worked pretty well. Sometimes the G doesn't work so well, but in majority they work. So now I'm designing some others, and I'm kind of a little getting confused. When you read about the CRISPR-Cas9, when you design your, because some programs gives you the strand, the gRNA, the strand that it reads and the PAM region, but it doesn't, or it's the opposite strand, right? And then your how can I explain? So if it's the opposite strand that you put it into the program, it will give you the, for example, ACC, right? So it's not the TGG, the PAM region, but it's ACC. So, but the program also can give you I'm using the CC top, and it works fine for me. It gives me the off targets, and I didn't have to go further, and it's okay for now. So some programs gives you the sequence of the gRNA for one strand, and then your PAM region is actually, if you put that gRNA as it is, the PAM is actually the opposite strand. And that happens usually with the gRNAs that are in the opposite direction of the strand that you put it in. Am I making myself clear? So based on this picture, this is what I'm confused. Based on this picture, your PAM has to be the opposite strand of your gRNA sequence. Is that correct?
No. So the NGG sequence is like the 20 nucleotides preceding the NGG are exactly the same because they're complementary to the other strand.
So your PAM region should follow basically the sequence and the strand of your gRNA.
Yeah. So the NGG, the 20 preceding nucleotides are exactly the same 20 that are in your guide RNA, your spacer that you're
Okay.
actually designing.
Right. All right. Okay. That answer, because I was thinking, but then I wasn't sure now based on this picture, because usually they present this picture like this, and then it's like, okay, what's going on? Why is this? Okay, I'm all good. Thank you.
Yeah, in the beginning, I used to have to refer to this picture more often than you would believe.
I mean, I refer to this as a plus and a minus strand. Like S strand is something that it follows my, the strand that I put it in, and then the reverse one is the antisense. So,
Yeah.
All right, thank you.
You're
Yeah,
welcome.
all are attending, please note there is no silly questions, so feel free to think about your question, write it in the chat, and also raise your hand. And so I'm gonna go with the question posed by David. Why all the guide RNA must start with G?
I can certainly answer that. They don't actually have to start with G. So the reason in this diagram that I had just shown, I think I'm not sharing anymore, but the reason that they very often do is because of the promoter we use to drive the guide RNA. So the guide RNA, most guide RNAs that we use, we're almost always using an RNA polymerase 3 promoter. And the one that we use the most in dicots is the Arabidopsis U6. So the transcription start site for that promoter is extremely well mapped. It starts at a G always. If you forget to put a G at the 5' end of your guide RNA, it won't start transcription until it reaches the first G in the guide. So your guide will be truncated in it. Depending upon when that G occurs, it may or may not work at all. So that's really important to know which promoter you're using. There's one that we use for it's like a rice one, I think. Erik probably knows better. But it actually doesn't start with a G. It starts transcription on, I think, an A, but I'd have to look it up because I don't use that one much.
Colby or Erik, one of you can address, do people who are interested in doing this, does it matter, or what are the considerations that they should think about if they're dealing with monocot versus dicot plants?
You want me to answer that, Colby?
Yeah, you do a lot more monocots, so.
Okay. if you're doing monocot work, you want to make sure that if you decide to use an RNA polymerase 3 promoter, that you use the one from rice, so the rice U6 or OS U6 promoter. That seems to work well in sorghum and Soteria and rice. But I think others have used it in different monocot species. But you don't always have to use a U6 promoter. In rice, in Soteria and sorghum, sometimes I use a Pol2 promoter to drive expression of guide RNAs, and that also works well. In that case, you don't have to use a guide that starts with a G. And then the other consideration you have to make between monocots and dicots is what type of Cas enzyme you're using. For dicots, we often use an Arabidopsis codon optimized Cas9 variant that has 2 nuclear localization signals on both sides of the gene. And in Soteria, we found that that works well, but it tends to work better when we co-express a 3' exonuclease known as TREX2 that can help increase deletion size and your gene editing frequency. So those are kind of the considerations I would make.
Thank you, Erik. Let's go with the person named Saurabh Pandey. If you can unmute yourself and ask your question. Let's see if we can unmute. If not, I'm gonna speak about his question. What specifications we need to consider when designing guide RNA editing polyploid genome or like banana?
Yeah, I mean, I can attempt to answer that one. We have worked in a few species that are reasonably redundant genomes. The most experience that I've had is actually working in wheat with this, and it was pretty painful actually to assay so designing the guides was actually not terribly hard, right? That's pretty straightforward. You line up all the homologs, and you either pick sequences that either target all the homologs that you want or only one of the homologs. You can kind of decide that's relatively straightforward to do that. It's basically, do you want to hit them all, or do you want to consider the other homologs? So for in wheat, like say you're doing the B and the D genome, you would consider those off targets, and so you kind of use all the same principles that you would use to say like I don't want to have an off target. But the part that actually I found the most difficult when working in wheat was amplifying specific genes, so that like to test actually whether or not I had made mutations in them. So what I tended to do is amplify all 3 genomes of the like homologs, which was not what I wanted. I wanted to be able to test every single one individually, and I would suggest that that's what you do want to do. And so when you're designing guide RNAs for very complicated or polyploid genomes, I think it's a very good idea to design the primers and test the primers. Very important to do the testing, not just the designing. The primers you're going to use to do your genotyping, and make sure that you can actually identify basically the single copy of the gene that you're actually trying to target, or as many of them individually. Because I did not do that, and it was very difficult. We basically kept getting all 3 genomes, and so then the sequencing and the analysis was actually way harder simply because of that. Like designing different guides would have been pretty easy, but we screwed that up, and we should have definitely done that differently.
There is one question. Thank you, Colby. And then there is a question from Rizki LN Wulan. It is about, so Colby, you touched on the methylation and then how it impacts the guide RNA efficiency, cutting efficiency. And so the question is, is there a way to find out if the guide RNA is located in unmethylated region using the guide RNA design website? And if you're answering, Erik or Colby, you are answering this question, so can you please also address what if there is no good quality sequence available? So what will be guidance you can give to people who are working on plant species, not good sequence available? And if it is available, what is the best tool to understand the methylation and finding the good guide RNA sequence?
That's a tough one, actually. I don't know terribly much about the databases that are available. Um, I definitely know that, like, for example, Arabidopsis has pretty good data, but generally, most plants don't have good data for basically where the methylated regions of the genome are, which makes this a challenging aspect of designing guides. And basically, at some level, it's just kind of a test and see. So the test can be done multiple ways, whether or not you're doing that by something like leaf infiltration or something like protoplast. For some species where transformation is actually relatively straightforward, sometimes you literally just transform the plant with multiple guides to find out. But there are going to be targets out there that people are interested in where there really is almost no chance that you're ever going to be able to do any editing with Cas9 based editing because like really we like it's so heavily methylated that the efficiency is effectively zero. And determining that is a challenge. It's unfortunately kind of a painful challenge, and I don't think that databases exist terribly. They're not great databases right now for especially, well, most plants. So
So basically, they have to do their own homework to get
yeah,
started
I
on
mean,
the right track.
yeah, I mean, you could try to basically sequence it. You do bisulfite sequencing of your locus, but I'm not sure that that's any easier than just like making a transgenic and testing. So.
Cool. There is a question hand raised by David. Can you please go ahead, unmute yourself and ask question?
So while going for gRNA design, we are considering that secondary structure and off targets. If the off targets are few, but secondary structure is very good, we can take that, or otherwise, uh, zero off targets with not bad secondary structure. Which one is more better, sir? We need to more focus on secondary structure or more focus on off targets. And one more, if both are good, but it is not start with G, we can use that or not. Can you understand, sir?
Yeah, I think so. So the first thing the easiest one to address is if it doesn't start with a G. So we have done things where we've added a 21st nucleotide at the 5' end of the spacer, um, and made that a G, and that does not seem to affect activity of Cas9 at all. So if it doesn't start with a G, we just add one. So that makes that very easy to answer. Then I think your other question was like, what's more important, off targets or secondary structure of your guide? So if I saw a guide that had a hairpin, like a pretty strong hairpin, either within the spacer region or with some other part of the scaffold of the guide RNA, like I would throw that out right away because the chances that like it would decrease the effectiveness of it. So it doesn't matter if there's off targets because it might not work on target, right? If there's on and off targets. And so as far as off targets, certainly, I pay attention to them, but at some level, we're working with plants. If you get multiple independent alleles of your knockouts or of your edited plants, the chances you get an off-target event that's exactly the same go down. And you can always cross the plant, so to back cross it into a wild type to help remove some of those off-target effects. It's certainly better if you can find one that has no or very few predicted off targets, but I don't really let that stop me when I'm designing guide RNAs. If there's a pretty close off target, but it's like exactly where I want the cuts to occur, I definitely use that guide.
Erik, you would like to add something on this topic?
No, Colby got it.
Okay, next question. I would like to pick up from the chat and then we will go with the people who are raising hand. It is from Payal Mawadia. How to design allele specific guide RNA?
So that's really it's kind of the same thing about talking about off targets. So typically, what I would do And kind of the most obvious example is something in a polyploid genome where you have like three copies of a single gene. So I would personally align them, and if there's one of them that you want to target specifically, what you want to do is the closer to PAM, the more important the mismatch is. So to say that and maybe in a different way, if there's a mismatch very close to the PAM between your spacer and kind of an off target, the less likely that's going to work. If you get close to the 5' end, sometimes like one or two mismatches don't matter. Like the guides, it doesn't seem to affect efficiency between the multiple target sites. Close to the PAM, or even the better, if your PAM is wrong, and that's your actual mismatch, that's kind of the best situation for trying to design ones that will target one sequence and not a very similar one.
Thank you, Kobe. There is a next question I'm picking from chat is from Joshua Paul Peselidula. What are the strategies to follow if I want to design a guide RNA at the promoter region of gene for promoter editing? What you would think people should be cautious about, careful, or how to go best?
Yeah, Erik, you go because you've done this.
Yeah, sure. So there's different Cas variants besides Cas9 that have different PAM preferences. So like Colby mentioned, Cas9 prefers NGG PAMs, which are often in gene bodies. If you want to target promoter sequences, people will often use a Cas variant known as LB-Cas12a, which has a T-rich PAM. And there are engineered variants that require TTV. So the V can be, I can't remember which two base pairs. But anyways, targets T-rich sequence. And LB-Cas12a will generate larger deletions than Cas9. So is there anything else I should add to that?
That should be good,
No?
Erik.
Okay.
Next question is from Quintin. Can you talk a little bit about the genotyping process to evaluate what type of edit was made? So just for all the attendees, I would like to say there is going to be help session 2 to address how to genotype, but there is a question which is relevant to this session is if I am looking to obtain a heterozygous knockout of a protein, as full knockout is lethal, unsure how, I would like to modify a little bit how you would go about designing the guide RNA. What will be your recommendation if the edit can cause a lethal outcome?
Yeah, so we don't know if we've ever encountered that. It's kind of hard to know until you basically prove it by actually just getting the het and then selfing it and seeing that you actually can never recover a homozygous mutant. Um, so it depends. A lot of that's going to be, I think, dependent upon your specific gene, right? If it's lethal almost immediately, so for example, it affects like the cell cycle or something like that, then the strategy is literally doesn't change because you're never going to recover. You're never going to see the homozygous mutant. Um, so if it's lethal, say it's sterile, then that is a little bit harder, but it's actually not terribly hard, at least in my experience, to recover heterozygotes. So that's not something we've ever struggled with terribly. If we can get a homozygous mutant, we can also almost always get a het. Um, even if my guide is working pretty well. And then to detect that, like Vina said, there's going to be this other session that's unscheduled at this point. But very, very briefly, what we usually use is either ice or what's the or tide to actually look at sequences. So that's basically you amplify your gene of interest, and then you just use Sanger sequencing to sequence across it, and then you can look at the trace, and you can actually pretty easily pick out a heterozygous plant from the Sanger sequencing trace.
Colby, can you say what a trace is? Just in case people don't know.
Yeah, I guess I can try. The sequencing trace. So that's basically the raw output from the dye terminator sequencing reactions, where you basically, if you aren't terribly familiar with Sanger sequencing, when you sequence it, what you end up with is basically kind of a fluorescence intensity for each of the nucleotides as it passes through a column. You know, it's excited by a laser, and you can actually see the intensity of the individual nucleotides. And so, if you have a heterozygous mutation, and most of the mutations we look at are either like +1 or -1 type of thing. You basically see this frame shift in that the sequence will look perfect. It'll be, all very nice, one nucleotide at each location as it comes off. And then when you hit the cut site and you have this now a heterogeneous population, it will actually have peaks of roughly equal size for two different nucleotides. And so it's very obvious. It looks like perfect sequence, and then suddenly it looks like your sequence has gone to hell because actually what's happened is you have one or sometimes more different kinds of sequences in the trace.
Thank you, Kobe. There is a question from Uma Shankar, and I'm going to modify a little bit because you have touched on this topic a couple of minutes ago. The question is, any suggestion on Cas and guide RNA design in sunflower? So how you can help address this is the crops where you do not have a good sequence available. Can you walk people on that path, how they can do or make most of their resources to make a good attempt on genome editing project, whatever they are working on? For the plants, you do not have good sequence available.
Yeah, so that's also a little difficult. We don't have a ton of experience with that because as people who actually are basically trying to develop new tools, we mostly don't work in crops or plants that don't have relatively good genomic resources. But if I'm doing that, the first thing is obviously you need to have the sequence of your target gene. Um, Probably, so for dicots, I would drive Cas9 with either 35S or something similar, basically a very strong promoter, but 35S has worked really well for anything that goes through tissue culture. Doesn't work very well for Arabidopsis, just in case you are unaware. And then for monocots, I would use the Maze ubiquitin to drive Cas9. I would always use the RNA polymerase 3 promoter to drive the guide RNAs. Whether or not that's the ATU6 or the RICE U6, either of those seem to work very well in either monocots or dicots, the respective one. And then if I didn't have great genomic sequence for the rest of it, but I did have my target gene, I would be sure to isolate multiple independent events. The reason why that's going to be important is because you really don't know what your off-target background is going to be unless you sequence the genome, and even then, to enough depth, that's going to be pretty difficult. And so if you have like 5 or 6 or more independent alleles, and then when you're phenotyping those, if they all have the same phenotype, you have increased your chances that the phenotype you see is actually dependent upon the gene that you've edited, that you've edited. It's still going to be, I could easily imagine a reviewer saying you're going to need to complement that mutant with a wild type copy of the gene because you don't know what else is going on in the genome.
Thank you, Kobe. Before we go to Quintin, there is a question I'm picking up from the chat. It is from Siddhant J. Bhai. The question is about how important it is to consider secondary structure for designing guide RNA.
Erik, have you thought about that a lot? Don't usually think about it too much.
I only think about it in the context of prime editing, but not when I design normal guide RNAs.
Got it.
And I think about if something fails, then that helps me make that decision.
Yeah.
So
Yeah, I think what I usually do look at the secondary structure is I literally look at it by eye, and I'm like, okay, well, I can see that there's like a bunch of G's and C's, and then I'm like, oh, I should probably check because it could well have like secondary structure, but it's not something I routinely look at. You're only adding 20 nucleotides to the sequence, and often it's in a coding sequence. The chances that you have like hairpins suddenly because of this I think, is probably pretty low. It probably is not a bad idea at all to check.
doing proper controls and just designing one RNA and it does guide RNA and it doesn't work, I think, doing a good experimental design will take people a long way. And Quentin, go ahead and ask your question. Unmute yourself and ask your question.
Hi. Thank you guys for hosting this. I wanted to ask what were the possibilities and what the process is like for using technologies such as prime editing to deliver short sequences to promoters of Arabidopsis genes. We're a bit interested in trying to insert very short enhancers in promoter regions, but I don't really come from a CRISPR background.
Yeah, I can answer this one. So I've done a little bit of that in rice, where we've inserted these 36 base pair enhancer sequences. And in rice, you can insert things up to 80 base pairs pretty efficiently and obtain, 20 to 30% of your plants will be heterozygous or homozygous for the edit. However, prime editing seems to be limited to mostly monocots. There's one paper published where they've attempted to do it in Arabidopsis, but they suffer pretty low efficiencies. And when I've tried to repeat their results, I don't have any luck. But if you wanted to try and do it a third time, they put their prime editing reagents and prime editing guide RNA sequences onto a Gemini viral replicon, which is a virus that has had its movement protein removed, so no longer moves through the plant, but it produces lots and lots and lots of copies of your gene editing reagents, so you boost your expression. And when they did this, they saw a slight increase in prime editing efficiency in Arabidopsis, but what really helped was to heat shock the plants several times after floral dipping. And that helps just because the reverse transcriptase fused to prime editor and Cas9 both have a temperature optimum of 37 degrees Celsius. So putting it at the correct temperature can really help boost prime editing efficiency.
So Erik, you mentioned high temperature, and so anytime we are doing or people are doing the transformation to deliver the guide RNA, the process is defined by the transformation or tissue culture process. And so what will be your recommendation when people thinking about this temperature requirement for the optimal efficiency of guide RNAs? It's very process-centric question,
Sure,
but
sure.
yeah.
Yeah, so like when you make Soteria cali or rice cali, those two plants have different temperature requirements when they go through tissue culture. Rice prefers 28 to 32 degrees Celsius, where Soteria likes 25 degrees Celsius. But in either case, I haven't observed any issues with heat shocking every 3 days for the 2 weeks that those plants are on their recovery medium after co-culturing with agrobacterium. They seem to handle it just fine. So I would imagine this extends to most other plant species, but you can always reduce the temperature of the heat shock if you're worried that 37 degrees Celsius will kill whatever you're working in.
And what is
But
the duration per heat shock?
I do 4 hours and then I recover it for 2 days at its preferred temperature before I do it again. And I'll do it 3 times normally.
Okay, thank you. There is a question by Risky. If you have an option to choose between U6 and U3 promoter, how you will make a decision to go about choosing one or the other?
So I can answer this pretty easily, and we actually published a paper in 2017 where we compare U6 and a U3. These are the Arabidopsis versions, and actually I think it was in that paper, but anyway, the U3s don't seem to work as well, basically. So we exclusively use the U6 promoters, and we've had very good success having many copies of the U6 promoter to drive individual guide RNAs. We only drive one guide per U6. And so I've personally done 24 different guides on a construct and had pretty good success. I know we've done in tomato something like 16 guides, all driven by an individual U6 promoter, and that works really well. So,
Say individual U6 promoter, it is one promoter driving 16 guides, or each guide driven by U6 promoter independent of each other.
yeah, so it's each guide is driven by its own U6. So we'll have 16 different U6 promote, like they're identical, but 16 copies. And then you basically have a repetitive, like very repetitive sequence. The only difference between each of the repeats is the spacer sequence in your guide RNA.
So spacer sequences are unique in nature.
Well, they're unique to your gene, your target.
Okay, got it. Okay,
Yep.
yeah. Okay, thank you. While I explore more questions, I wonder if each one of you, Colby and Erik, if you can speak about situation. I wish I knew this and I would have saved myself from the pain and suffering. So, what will be one or two things you think it is good for everybody to know?
Okay, I can go first because that way Erik can't use mine. And that is so I actually watched on Plant Gene one of the kind of one of these expert master classes, and Tom Jacobs was talking about this, and Tom highly recommended that you design your genotyping sequence primers and test them and sequence your products before you actually even design your constructs. Um, we don't typically do that. Nobody does that, as far as I know, except maybe the people in Tom's lab. And I would say that is some extremely good advice. I still am not very good at that because, you kind of just like, okay, let's go ahead and make the construct, and then I'll work on the guide RNAs or the sequencing primers while it's in tissue culture, while we're doing the transformation. And inevitably, we get the plants out the other end, and we're still working on those stupid sequences, the primer sequences for genotyping. So I recommend doing that ahead of time, or at least at the same time.
So, knowing how to screen is also equally important as choosing the best guide.
Yes.
How about you,
Yeah.
Erik?
Sure. I would say I've done a lot of stuff in Soteria, and one of the mistakes that I made early on is using the wrong reference genome to design my guides, which is similar to what Colby said. So make sure you're looking at the correct reference genome because you have lots of differences between your two cultivars, which will drive your efficiency down. Then the second thing I'll say is make sure to do a little research on which promoters work best in your plant. Oh.
One thing I would like to add that when you are designing guide RNAs and it is based on some reference sequence, so it is always good to check your actual material you are going to work on to check the presence of the same sequence, because it can lead to wrong direction. You are making an assumption the seed look like this should be matching the reference genome. So that is also very important, that your starting material is accurate so that you can troubleshoot if needed or something goes south. Question from Shajneen is how to check guide RNA efficiency.
There's a couple of ways you can do it. The most thorough way that you can do this is literally either infect your plant in some way. So that can be protoplast, that can be it can be leaf infiltration. There's a bunch of ways that basically you get the reagents into the plant. It could even just be like taking your plants to callus, infecting with agrobacterium, and then doing a genomic DNA prep. But then basically high throughput sequencing across the target site. So amplify that target site and do high throughput sequencing. That is by far the most accurate and effective way to do it. There's some potential problems with something like protoplast because you end up delivering so much of the reagent, but you at least would know that it worked. I think that the percentages, say you get 60% editing if you try in protoplast, that might not be truly reflective of what you're going to see when you try to do whole plant transformation, because again, you can deliver so much of the plasmid DNA to the cells. But it will give you kind of a yes no. Like if it doesn't work in protoplasts, it's probably not going to work in your tissue culture plants. And that gives you, as far as relative amount, if you're trying to test multiple guides, that's going to give you a pretty good readout. That's also kind of expensive and a pain in the butt. Sometimes getting those data back takes like 6 weeks, something like that. A relatively straightforward system would be to do kind of the same technique, except that you do the Sanger sequencing, and then you use either Tide or ICE. Again, these are those programs that allow you to deconvolute the trace of the Sanger sequencing to give you a rough estimate of frequency. I all those estimates that you get from either Tide or ICE are, in my opinion, plus or minus about 10% editing efficiency. And really anything below 5% we don't believe. That's probably just noise in the sequencing reaction. So, Vina, you're muted.
Question is from Sri Kurniati. How many nucleotide distance between two guide RNAs to make both work? Like what will be the best optimal recommendation or the range you could recommend?
Now, Erik, can you do this one? I think you've done the most on this.
Sure. So I usually set the minimum distance between both guides as the size easiest to see when you would run your DNA band on a gel. So I would say that's like 50 to 100 base pairs. Because if you get a deletion that size, then you don't always have to Sanger sequence or use NGS to sequence your target site. You can just look for a shift on the gel between the wild type and your deleted thing. That being said, we've made deletions that span from 100 to 200 base pairs using 2 to 4 guide RNAs, but generally I aim for guides that are spaced 50 to 100 base pairs apart in the PAM inward orientation.
The question is the heat shock better done at Calais stage versus selection or later stage?
The heat shock seems to work better at the callus stage. So after you've co-cultured with agrobacterium.
So have you, Erik, have you ever tried inducible promoter for the guides?
Oh no, I've never tried inducible promoters for guides.
Colby, did you?
No, not for guides or the Cas. We used to do that a long time ago when we used zinc finger nucleases and talons, but mostly because we were worried that we were going to chop up the genome and basically make the plants like it would basically kill them. They wouldn't be able to go through cell division because of checkpoint control. But we never actually found that to be the case. So I think there might be some reasons why you would do it, but they're more complicated. Having to do with tissue specificity or embryo lethality or those kinds of things. But that's not really anything to do with the guide RNA design.
Thank you. And we are nearing very close to the end, I believe, but we are going to continue with the question until we are moved to the main room. One question, both Colby and Erik, you have analyzed a lot of plants, and then I'm very confident we are going to cover it in the next session. And it is about when somebody is generating the plants. Do people care or should they care about number of copies of the tDNA, high copy better versus low copy, or what you would advise people on that topic?
We just had a whole meeting about this like yesterday.
Yeah.
My personal opinion, and Colby can disagree with me if he likes, is that copy number doesn't really matter. What matters is how many insertion sites you have. So you could have hundreds of repeats of the tDNA in tandem next to each other. And as long as there's only one insertion of that, you can segregate away the Cas9 gene if that's your intended goal pretty easily. I don't know if you want to add more to that, Colby.
Yeah, I would say, in primary analysis, I don't even care if there's 3 or 4 or 5 insertion sites. It's really about your downstream phenotyping and basically trying to get things published. And especially if you're trying to work in a crop, and at least in the US, and you want what's called a null segregant, right? You want any transgenes segregated away. The more insertion sites you have, that can be just basically a longer process. Or if, God forbid, you get one that lands really close to your edit of interest, and then segregating it away is really, really hard. But like generally for primary analysis, multiple insertions, like either in tandem, as Erik mentioned, or even in different sites, that doesn't really bother me too much.
Yeah, and I'll just say in Soteria, we've edited like probably 12 different things where we've just back crossed it once and we were able to obtain Cas9 free plants. So
So
happy number.
in your experience, whenever there is high copies inserted, whether single locus or multiple locus, did you see high chances of edit, or single copy is equally good if it is located in expression, like expressing region of the chromosome?
We don't really sequence the whole genome all the time, so I don't think we have the tools to answer that question. But we just know from like digital droplet PCR or QRT PCR that there are lots of copies generally.
Yeah, but doing the correlation between the number of insertion sites, it requires finer data than we have. So
Yeah.
we don't know.
Thank you. There is a question from Likindra. Question is how to design polycistronic tRNA cis4 guide RNA for multiple target editing in plants with a single construct.
So let's see. I'm trying to remember. We have like a protocols paper on how to do that. I made a system that allows us to clone it. The actual design of the guide RNAs is really no different than designing a single guide. The cloning is a little bit more complicated. There is a protocols paper. The first author is John Cody out of our lab. It's like a Nature's protocol. And I think it's buried in there. That's not the focus of the main paper. And all those reagents are available at AdGene. So if you're interested in that, I would say look there. But the actual guide design is really not any different.
Thank you. Next question from Julie Thakur. How is the position of an exon 1, 2, or 3 important for guide RNA design, especially when the first exon information is not available?
Okay, so I can try to answer that one. Let's see. So basically, what we tend to think is the closer to start, the more likely that a single frame shift is going to cause a null mutation, which is what most people are looking for, is a null. If you go further into the gene and you actually have, for example, a functional domain that is correctly translated, you might think you have a null, but you could have either a relatively wild type phenotype or gene function, or you could even have a hypomorph where you've actually changed the function of the resulting protein, which is complicated and probably a giant pain to figure out. So that's kind of why we end up going closer to the 5' end of the gene.
Thank you. We have only 7 seconds to go. I would like to thank you all and Colby and Erik for joining us for this help session.